Hello, everyone. You are welcome to my YouTube channel. In continuation of the discussion of the basic microbiological tests or protocols, I'll be discussing the characterization of Staphylococcus aureus. Staphylococcus aureus is a gram-positive coccus, which is non-sporing, or it is a non-spore-forming organism. It is non-motile, and the cell arrangement under the microscope is the grape-like clusters, or the cells are arranged like a bunch of grapes. Most staphylococci are normal flora of the skin and mucous membranes of humans, and they hardly cause infections when they are found on the intact skin. However, if there's a break in the skin, Staphylococcus aureus in particular will produce infections and diseases. Staphylococcus aureus is known to produce an array of enzymes such as coagulase and toxins to enhance their pathogenicity. Their pathogenicity, that is referring to their ability to cause disease. Staphylococcus aureus is known to cause acute endocarditis, post-operative wound infections with abscess formation, fetal septicemia, myocarditis, suppuration, and mastitis. There are several biochemical tests that are used to identify Staphylococcus aureus. The first one is the coagulase test. The coagulase test involves two tests, the slide coagulase test and the tube coagulase test. The procedure for the slide coagulase test includes the following. A colony of the test organism, in this case Staphylococcus aureus, is emulsified carefully in physiological saline, and a drop of citrated rapid plasma is added and mixed on a microscope slide. Slide coagulase test detects bound coagulase. So if bound coagulase is present on the bacterial cells, then the citrated plasma will cause the bacterial cells to clump. Clumping will occur because the clumping factor is an adhesin, which causes the cells to bind to fibrinogen in the plasma. The slide coagulase test is a very quick procedure and is performed first to detect the bound coagulase, and only a negative strain of Staphylococcus aureus is further tested in the tube coagulase test. If a positive result is obtained, the organism is reported as coagulase positive Staphylococcus aureus. The coagulase test differentiates strains of Staphylococcus aureus from coagulase negative Staphylococcus species such as Staphylococcus epidermidis. The second type of coagulase test is the tube coagulase test, and is performed by emulsifying 2 or 3 colonies of the pure culture of the test organism in 1 ml of citrated plasma in a serological tube. As the bacteria multiply in the plasma, they secrete coagulase. What is coagulase? The coagulase is an enzyme which initiates blood coagulation by activating prothrombin. And it causes the blood coagulation to occur, which is quite different from the normal blood clotting process. So coagulase binds to fibrinogen, forming a complex, and this cleaves the fibrinogen, which is soluble, into the insoluble fibrin clot, which can be seen. Here is a plate of a positive tube test reaction. The insoluble fibrin clot can be seen here, while plate 2 shows the negative coagulase tube test reaction, which means the soluble fibrinogen is still in solution. There is no clot here. It is important to note that if an organism is able to produce fibrinolysin, it will digest the fibrin clot. So the incubation should not be done for too long. If it is for so long, this fibrin clot will be dissolved, and this will give a false negative coagulase tube test. The second test that should be done, or that can be used to identify Staphylococcus aureus, is the catalase test. Again, there are two types of catalase tests. You have the slide catalase test and you have the tube catalase test. Catalase is an enzyme which is present in all human cells, plant cells, animal cells, microbial cells, except Streptococcus species. This test is essential for differentiating catalase positive Micrococcaceae from the coagulase negative Streptococcaceae. The procedure or protocol involves placing a microscope slide inside the petri dish. Although the use of a petri dish is optional, as the slide catalase can properly be performed without it. However, the use of a petri dish is strongly recommended. So using a sterile inoculating loop, a small amount of the test organism from a well-isolated 18 to 24-hour colony is picked and is placed onto the microscope slide. It is important not to pick any agar along with the colony. This is particularly important if the isolate has been grown on agar containing red blood cells. Like I told you before, human cells contain catalase. So if agar, for example, blood agar, is picked along with the colony, this may result in a false positive reaction. Using a dropper or partial pipette, place 1 drop of 3% hydrogen peroxide onto the organism on the microscope slide and do not mix. Immediately cover the petri dish with a lid to limit aerosols and observe for immediate bubble formation or effervescence. Observing for the formation of bubbles against a dark background enhances readability. Positive reactions are evident by immediate effervescence, that is bubble formation. Here you have a plate showing the slide catalase test result. The top part shows the positive reaction produced by Staphylococcus aureus, while the negative reaction at the bottom is produced by Streptococcus pyogenes. It is important to note that the reaction is immediate. You have the immediate bubbling and effervescence for Staphylococcus aureus. The second type of the catalase test is the tube catalase test. Here, 4 to 5 drops of 3% hydrogen peroxide is placed in a test tube, and using a wooden applicator stick, a small amount of the organism from a well-isolated 18 to 24-hour colony is picked and placed in the test tube. It is also important not to pick any agar, particularly blood agar. This is important because the isolate grown on agar containing red blood cells or blood agar may give a false positive reaction. Now, the tube should be placed against a dark background and observed for immediate bubble formation. Here is a plate showing the positive test catalase, a positive tube catalase test. Here you can see the bubbles that are formed, the immediate effervescence. Here is a plate. Plate 5 shows the negative tube catalase test. There is no evidence of bubble formation or effervescence here. If an alternative is required, the test organism can be grown on agar slant, and so directly a few drops of hydrogen peroxide can be added to the surface of the agar slant. Here you have evidence of effervescence bubble formation on the agar slant where the organism has been grown. Plate 7 shows the negative tube slant catalase test. There is no evidence of bubble formation or effervescence on the surface of the agar. All these images the source of the images used in this video is the American Society for Microbiology Microbe Library. The third biochemical test which can be done to identify Staphylococcus aureus is gelatin hydrolysis test. The gelatin hydrolysis test detects the ability of bacteria to produce the enzyme gelatinase. This test distinguishes the gelatinase positive pathogenic Staphylococcus aureus from the gelatinase negative non-pathogenic Staphylococcus epidermidis. And gelatinases are proteases actually secreted extracellularly by some bacteria, which hydrolyze or digest gelatin. There are two types of detecting gelatin hydrolysis. The first one is the nutrient gelatin stab method, which is the standard and most commonly employed method. It involves the inoculation of the nutrient gelatin tube by stabbing the test organism into the butt of the tube, and then the tube is incubated at 37 degrees Celsius for 24 hours. After 24 hours, the nutrient gelatin tube is transferred to a refrigerator and removed after 30 minutes to 1 hour, and then examined for evidence of liquefaction. If there is no liquefaction, the tube should be returned to the incubator for another 24 hours and observe daily for liquefaction after refrigeration. This is done for up to 7 days. Here is a plate showing the positive gelatin liquefaction test. See the evidence of gelatin liquefaction here, and it shows that the test organism has produced gelatinase, which hydrolyze the gelatin liquid, leading to liquefaction. Here is plate 6 showing the negative gelatin liquefaction test. The test organism was not able to elaborate the enzyme gelatinase, and so following refrigeration, incubation and refrigeration, the gelatin is still intact and is not liquefied. The second type of gelatin hydrolysis test is the nutrient gelatin plate method. Here, a heavy inoculum of an 24-hour test organism, presumptive Staphylococcus aureus, is inoculated onto culture plates that have been pre-filled with nutrient gelatin. These plates are incubated at 37 degrees Celsius for 24 hours. Gelatin hydrolysis is indicated by clear zones around the gelatinase positive colonies, following the flooding of the gelatin plates with either acidic mercuric chloride or, in some cases, saturated ammonium sulfate. These reagents will precipitate unhydrolyzed gelatin, making the clear zones easier to see. Results can be observed within 5 to 10 minutes after flooding with saturated ammonium sulfate. Here is a plate showing the nutrient gelatin plate method. The test organism isolate A, which was inoculated here, and following flooding with saturated ammonium sulfate, shows evidence of precipitation of the gelatin in the rest of the plate, while you have a clear area of gelatin hydrolysis around the colony. Then isolate B, which is the second test organism, it could not produce gelatinase, the enzyme gelatinase. There is no evidence of clearing clearing around the colony, showing that it did not produce gelatinase. The fourth biochemical test which can be used to identify Staphylococcus aureus is the DNA test. Here, the procedure simply involved the inoculation of DNA agar plates simply by streaking a colony obtained from the culture of presumptive Staphylococcus aureus. The plate should be incubated at 37 degrees Celsius for 24 to 48 hours. After incubation, the surface of the DNA agar is flooded with 10% hydrochloric acid. If the test organism is able to elaborate the enzyme DNase, the enzyme will digest the DNA in the immediate vicinity of the streak, and a positive test is indicated by a clear region around the growth of the test organism. This can be seen in this plate. Isolate A and B show evidence of a clear area around the streak where the test organisms were inoculated on the surface of DNase agar. Here you have the clear area around isolate A and isolate B, following the flooding of the DNA agar after incubation. While for isolate C, there is no clearing there is no clearing around the streak, meaning that isolate C did not produce DNAs. Another test which can be done to identify Staphylococcus aureus are the carbohydrate fermentation test. Staphylococcus aureus is known to ferment the following sugars: mannose, glucose, galactose, fructose, and mannitol. However, it does not ferment raffinose and xylose. Another test which can be used is the beta hemolysis on blood agar to identify Staphylococcus aureus. Beta hemolysis is complete hemolysis characterized by a clear zone around the colonies on blood agar. There are rapid identification protocols such as thermoscientific RAP-ID Staph Plus system, immunological procedures such as enzyme-linked immunosuppressant assay ELISA, immunoprecipitation, and also immunoblotting. Also the hologic AccuProbe and PCR application of 16S rRNA gene can be used as rapid identification protocols for Staphylococcus aureus. Here are some selected references and links for further reading. Thank you for watching.